SNP discovery and SNP genotyping. There are over 1 million SNPs identified. 95% of these are unique and valid polymorphisms. Method for SNP discovery or detection involve a set of biochemical reactions that isolate the precise location of the suspected SNP and then directly determines the identity of the SNP using an enzyme called DNA polymerase. Also, many SNPs were initially detected by comparing different sequenced genomes. The second phase of human genomics, the first being the sequencing of the human genome, involves a large-scale screening of different human populations for significant DNA polymorphisms. High-throughput SNP genotyping is the process of quickly and cost-effectively identifying the SNP values in as many different individual human genomes as possible. Steps of SNP genotyping involve DNA sample preparation, PCR amplifications, and microarray assays. The microarray technology must label SNP locations of both alleles in the DNA sample and determine the base values. ORCID biocomputer and Affymetrix are the leaders in providing SNP genotyping technology. They have developed single nucleotide polymorphism genotyping assays that combine ORCID's proprietary GBA primer extension technology with an Affymetrix gene chip universal array. Their technologies can provide ultra-high throughput 1 lakh genotypes per day. It is interesting to note that nearly all SNP genotyping uses Affymetrix equipment including GenFlex Affymetrix universal microarray. High throughput SNP genotyping achieves the goal of recording the SNP location base values of thousands of people from a given population. The genotype data gathered from high throughput genotyping is then used to determine the related haplotypes. Pharmacogenomics. Some SNPs are associated with the metabolism of different drugs. The association of a wide range of human diseases like cancer, infectious diseases like AIDS, leprosy, hepatitis, etc., autoimmune, neuropsychiatric and many other diseases with different SNPs can be made as relevant pharmacogenomic targets for drug therapy. Pharmacogenomics is a study of how variations in the human genome affect the response to drugs. The older term pharmacogenetics was derived from the words pharmacology, the science of drugs, and the genetics, the study of genes and their functions, to indicate the intersection of the pharmaceuticals and genetics. The newer terms pharmacogenomics describes such large scale, often genome-wide approaches. The sequencing of the human genome and the introduction of new technologies have made it possible to analyze multiple genes simultaneously. Pharmacogenomics may permit drugs to be tailor-made for individuals and adapted to each person's own genetic makeup. Diet, age, lifestyles, environment and health can influence a person's response to medicines, but understanding an individual's genetic makeup is thought to be the key to creating personalized drugs with greater efficacy and safety. Pharmacogenomics combines traditional pharmaceutical sciences such as biochemistry with annotated knowledge of genes, proteins and single nucleotide polymorphisms. A single SNP may cause a Mendelian disease, though for complex diseases not usually function individually. All types of SNPs can have an observable phenotype or disease. SNPs is non-coding regions can manifest in a higher risk of cancer and may affect mRNA structure and disease susceptibility. Non-coding SNPs can also alter the level of expression of a gene as an EQTL, that is expression quantitative trait locus. SNPs in coding regions do not result in a change of amino acid in the protein, but still can affect its function in other ways. An example is the silent mutations in the MDR1 gene, that is multi-drug resistance gene 1, which codes for a cellular membrane pump that expels drug from the cell, can slow down the translation and allow the peptide chain to fold into an unusual conformation, causing the mutant pump to be less functional. In MDR1 protein example, C1236T polymorphism changes a GGC codon to GGT at amino acid position 412 of the polypeptide, both encode glycine, and the C3435T polymorphism changes ATC to ATT position in 1145, both encode isoleucine. What pharmacogenomics testing does? The purpose of pharmacogenomic testing is to find out if a medication is right for you. A small blood or saliva sample can help determine whether a medication may be an effective treatment for you, what best dose of a medication is for you, whether you could have serious side effects from a medication. Current limitations of pharmacogenomic test. It include one single pharmacogenomic test cannot be used to determine how you will respond to all medications. You may need more than one pharmacogenomic test if you are taking more than one medication. Pharmacogenomic tests are not available for all medications. Because pharmacogenomics tests are available only for certain medications, your healthcare provider determines if you need to have a pharmacogenomic test prior to beginning of specific treatment. There are currently no pharmacogenomic tests for aspirin and many over-the-counter pain relievers. Pharmacogenomics testing costs and coverage. The cost of pharmacogenomics testing varies depending on which test is ordered and your health insurance coverage. A federal law called the Genetic Information Non-Discrimination Act (GINA) generally makes it illegal for health insurance companies to discriminate against you based on your genetic information. This federal law does not protect you against genetic discrimination by life insurance, disability insurance or long-term care insurance companies.